Namaskar everyone, welcome to this channel. So today I'm going to present on the topic nucleic acid, its structure and as genetic material. So in today's content we will discuss about nucleic acid as genetic material and the structure of nucleic acid. Nucleic acid. What is nucleic acid? They are naturally occurring chemical compounds that serves as main information carrying molecules of the cell. So chromosome mainly consists of nucleoproteins, and these nucleoproteins have two components, nucleic acid and other is the protein. There, there are the following experiments that were involved in demonstration of the nucleic acid and contains genetic material. First was the transformation principle that was given by Griffith in 1928. Second was the bacteriophage infection by Hershey and Chase in 1952. And the third was the tobacco mosaic virus infection by Frankel and Conrad. So here is the structure of a DNA deoxyribonucleic acid and RNA ribonucleic acid. Here adenine, thymine, cytosine, and guanine are the base pairs. And here, instead of thymine, uracil. Transformation principle of Griffith that was given in 1928. Griffith inserted two strains of Diplococcus pneumonia into the mice. One was the rough strain that was non-virulent. It was injected in the mice and that survived. The second was the smooth strain that was virulent, injected, and the mouse died. And the third was the heat killed smooth strain and the mouse survived. And the last case was the rough strain along with the heat killed smooth strain, the mouse died. So it was restudied, or we can say repeated by O.D. Avery and C.M. MacLeod and Sikarthi in an in vitro system. So they did this to identify the transforming substances that are responsible for converting the non-virulent into the virulent type. Virulence in Pneumococcus depends on the polysaccharide capsule, which was present in the virulent strain of S3 and was absent in the rough strain, that is R2, that is non-virulent. So the cells of non-capsulated rough strain were treated with the extract of DNA from capsulated smooth strain. So few cells of smooth strain type could be isolated from the mixture. The phenomena of transforming the characters from one strain to another using the DNA extractor was called as transformation. So when extract treated with DNase is the enzyme that destroys the DNA. So transformation was lost. But when protease was used, it did not affect the transformation. So protease are the enzymes. So protease are the enzymes that destroy the proteins. So here we can say that the DNA was absent, that's why the transformation lost. So DNA was the genetic material, not the proteins. Bacteriophage, T2 phase. Bacteriophage are actually the viruses that affect the or infect the bacterial cells. It consists of head and the tail. The head consists of protein coat and DNA. Here in the picture you can see this is the head. The cover is the capsid. Inside genome is present. Here is the collar and this is the tail region. Tail region consists of the tail core, tail sheath, and the last is the tail fiber. The phage attaches to the host with the help of these tail fibers. Hash and chess experiment. So it was conducted in 1952, and it was to demonstrate that DNA of phage enters the host cell. So DNA of bacteriophage is labeled with 32 isotope of phosphorus, and protein was labeled with 35 isotope of sulfur. So DNA was made radioactive by growing infected bacteria in the medium of P32 and O4, and the phage proteins didn't contain the phosphorus. Only DNA will be labeled. DNA didn't get contained with the sulfur. The infected bacteria was agitated in the blender, and after shaking, what we got is that the P32 was associated with the bacterial cells, but S35 was found only in the surrounding area. So the DNA entering the host cells carries all the genetic information for synthesis of new phase particles. So we can say that nucleic acids are the molecules of heredity. DNA is the sole genetic material in the DNA bacteriophage. Now TMV infection. That is tobacco mosaic virus. TMV do not contain DNA. It is actually composed of 6% RNA that is surrounded by a cylinder of protein subunits. Here you can see. So experiment was actually conducted by Frankel and Conrad in California to show that RNA is genetic material in the viruses. Frankel-Conrad experiment on tobacco mosaic virus. So two viruses were used. That was tobacco mosaic virus, and other was Holmes ribgrass virus. This Holmes ribgrass virus (HRV). HRV is isolated from Plantago lanceolate. So proteins of these two viruses actually differ in frequencies, different sequences of amino acid, and their symptoms are also different. So TMV produces molting, whereas HRV produces distinct rings. The reciprocal chimeras using RNA of one strain and protein of other strain could be obtained. Chimeras used for infection showed the progeny had proteins which corresponded to the virus, which RNA of infecting virus particles was derived. So after demonstration of this genetic nature of RNA in TMV, RNA was found in number of bacteriophages. So it actually proves the specificity of virus proteins was determined by RNA alone, and the proteins carried no information. Structure of nucleic acids. Nucleic acid is a long chain polymer composed of monomeric units, means single units, nucleotides. Nucleotides are combination of nucleoside plus phosphate group. Nucleoside is base, that is nitrogenous base plus pentose sugar. In case of RNA, sugar is ribose, and in case of DNA, it is deoxyribose. So what is the difference is that in the C2 position, it is hydroxyl group and ribose, and here hydrogen in C2 position of deoxyribose. So nitrogenous base are heterocyclic and planar, and they are also not soluble aromatic molecules. There are two types of bases, pyrimidine and purines. Purines are found in DNA, like adenine and guanine. Adenine has NH2 amino group at cis exposition. At cis exposition, in guanine it is C2 position. In DNA, thymine and cytosine, whereas in RNA it is uracil and cytosine. Okay, instead of thymine, it is uracil in RNA. If we talk about the pyrimidine, cytosine contains NH2 group at C4 position. Uracil and thymine structure are same. Almost it is similar, but here what is the difference is at the C5 position, CH3 is there. Okay, in thymine, but in case of uracil, it lacks CH3 group, methyl group. Nucleosides. So nucleosides, the bond between the sugar and the base is called as N-glycosidic bond. The nitrogenous base lies above the plane of the sugar. That is the configuration of nitrogen glycosidic linkages beta glycosidic bond. Here you can see the pentose sugar and nitrogenous base is linked the position of 9 in case of purine and position of 3 in case of pyrimidine. The base is free to rotate around glycosidic bond, hence gives two different conformation, that is syn and anti. Okay, anti conformation actually it means that the base is rotated away from the sugar, and this is mostly favored. But in case of syn conformation, the purine base rotated towards the sugar. It is very less common. Okay, uh, purines have both syn and anti conformation, but in case of pyrimidine, only and only anti conformation. So this is due to the steric hindrance or steric interference between the oxygen at C2 position and C5 position. Actually, pyrimidines are large and close to sugar when they are in syn conformation. So this leads to steric repulsion. It causes crowdy. So that's why uh, syn conformation is not preferred. So what are the functions of nucleotides? First is they are responsible for the formation of polynucleotides of the DNA and RNA. And polynucleotides are informational macromolecules. They store, transmit and translate the genetic information. And they also acts as the precursor for the important compounds to participate in various metabolic reactions. Structure of DNA. So, who discovered the structure of DNA? So it is generally said that Francis Crick and Watson have discovered the structure of DNA, but surprisingly, it was discovered by Rosalind Franklin through her X-ray diffraction images, particularly the photo 51. So here is the photo 51. It was taken by a student of Rosalind Franklin, and it shows the B form of DNA, and she established the relation of double helix structure of her DNA. Her work revealed the density of DNA, showed that it exists in a helical structure, and this laid the foundation for the James Watson and Francis Crick model in 1953. She discovered the structure of DNA in 1952. While Watson and Crick are credited with the discovery, and Franklin's data, including this photo 51, provided key site that enabled their breakthrough. While the official announcement of the double helix model by Watson Crick was made in 1953, Franklin's work was fundamental and it was already discovered in 1952. But she was mainly focused on the A form of DNA. That is because of the crystallography, the change or variation in the humidity of the structure of the DNA. So, for more information, I will attach this link below and you can go through it and read briefly about it. The structure of DNA. So there are some generalization according to Irwin Chargaff. Here is the photo of Irwin Chargaff. That adenine is equal to thymine, guanine is equal to cytosine, A plus D by G plus C is equal to 1. DNA was also studied by Wilkins through X-ray crystallography, and he demonstrated that DNA was having a helical structure with diameter of 34 angstrom. DNA consists of double helix, two polynucleotide chains coiled on same axis. Bases are aligned in a plane that is to the right angle. Two polynucleotide chains run in opposite direction to have complementary base sequence. The distance between two base pairs is 3.4 angstrom and there are 10 base pairs at each turn and 20 angstrom in molecule is the constant diameter. So here we have to remember that when two purines are together, it would require a huge space, but in case of two pyrimidine, it would occupy very little space. When purine with pyrimidine pairs up, the space remains constant. Okay, so the alternative forms of DNA. So it varies due to the number of residue per turn and number of turns, spacing of residues along the helical axis H. There are three forms of DNA: aDNA, bDNA, and zDNA. A form of DNA. So A form of DNA is right-handed double helix made up of deoxyribonucleotides. It appears when relative humidity of environment is less than 75%. So this line is the only region that the Rosalind Franklin was interested in A DNA form instead of B DNA form. Two strands are anti-parallel with each other and not symmetrical. And this aDNA form is rarely present in normal physiological condition. So the molecule is asymmetrical because the glycosidic bond of a base pair are not diametrically opposite to each other. Major grooves and minor grooves can be observed in each turn. One turn of helix consists of 11 base pair with a length of 2.86 millimeter. In case of bDNA, it is 10 base pairs in one turn. Okay, so backbone of a DNA, aDNA, is formed by sugar phosphate that are linked continuously using phosphodiester bonds. All the nitrogenous base are at the core center of the helix. Hydrogen bonds between nitrogenous base allow the molecules to exhibit double helix structure. So That's why we can say that the A DNA is wider than the B DNA. So helix width is 2.3 nanometer. BDNA form. So this is the structure of BDNA form. It is also right-handed double helix discovered based on the X-ray diffraction pattern. It is the common form of DNA exists under normal physiological condition. The double strand of BDNA run in opposite direction. So this is also asymmetrical. It is the same reason that is due to the glycosidic bonds of base pair are not diametrically opposite to each other and have major grooves and minor grooves present alternatively in one turn 10 base pairs with length of 3.4 nanometer. The distance between the adjacent deoxyribonucleotides is 0.34 nanometer. It is same as the aDNA. The backbone of bDNA is formed by sugar phosphate that are linked continuously with phosphodiester bonds, and the core region consists of the nitrogenous bases. The helix width of bDNA is 2 nanometer. So this is the Z-DNA form. It is left-handed double helix and has a zigzag appearance of the backbone, which allows it to be distinguished from other forms of DNA. So it has a very different structure when we compare with the A and the B DNA. Okay, so the helix width is 1.8 nanometer, that is narrowest among all the three forms of DNA. The structure consists of major and minor grooves. One ton of Z DNA has 12 base pairs and the length is 4.56 nanometer. The distance between two adjacent deoxyribonucleotides is 0.37 nanometer. ZDNA is difficult to observe since it is unstable, but it has same as other forms of DNA. It has a hydrogen bond present to hold the two strands of DNA together. It may take part in expression regulation of some genes and other genetic recombinations. Okay, it can be found in bacteria, eukaryotes, and viruses. In some viruses, they require zDNA binding proteins for pathogenesis. So this is the structure of zDNA form. Now let's talk about the structure of RNA. Okay, so it is a single-stranded biopolymer of ribonucleotides that is bonded with each other through phosphodiester bond. RNA strand is synthesized in 5' to 3' direction from a locally single-stranded region of DNA. So it has ribose sugar that are attached to four bases: adenine, guanine, uracil, and cytosine. So ribose sugar has extra hydroxyl group in 2 carbon position. So this extra hydroxyl group in RNA led them to be synthesized for short-term functions. So three-dimensional structure of RNA is critical to its stability function. This is the structure of RNA. RNA is single-stranded and can form a complex structure by allowing its ribose sugars and bases to be modified on the action of cellular enzymes to perform various functions. Okay, and they even are capable of folding themselves, means into them, and showing intramolecular hydrogen bonding between the complementary strands, making them double-stranded molecule to exhibit specific function. So these are the types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. This is the messenger RNA, and this is the structure. It includes untranslated region, open reading frame, polyadenylated tail. So it is a single stranded RNA molecule that is complementary to one of the strands of DNA. mRNA are actually a group of RNA that can be translated into proteins while other RNAs cannot. mRNA is version of genetic materials that leave the nucleus and move to the cytoplasm where proteins are to be synthesized. When ribosome moves along this mRNA, it reads the base sequences using genetic codes, that is the codons in the form of triplets. Okay, it was given by Hargobind Khurana. So the structure of mRNA consists of uh, 5' end and coding region, 3' untranslated region, poly A tail. Okay, uh, and if we talk about the function of mRNA, it plays an important role in cellular functions, transcribing genetic information from DNA and guiding the amino acid sequence on the protein chain in the ribosomes. Okay, in prokaryotes, it contains a transcribed copy of DNA sequence with a terminal 5' triphosphate group and 3 hydroxyl residue. In eukaryotes, 5' triphosphate residue is esterified, forming a cap structure. 3' ends contain a poly A tail that consists of multiple adenosine monophosphate, that is AMP, and is added enzymatically after transcription. mRNA consists of intrones and exones and are subjected to process of cleavage. Okay, so in case of prokaryotes, it lacks poly A tail and a cap structure. So that's why mRNA of prokaryotes is less stable as compared to the um, eukaryotes. And one more thing, we can say that mRNA molecule that uses genetic code present on the portion of DNA makes the protein. If mRNA would not exist, then what would happen? The information on DNA could never be used. So that's the role of mRNA. So this is rRNA, that is ribosomal RNA. It is a type of stable RNA that is major constituent of ribosomes. rRNA consists of two subunits. This is the large subunit and this is the small subunit. Ribosome Ribosomal RNA acts as ribozyme, catalyzing peptide bond formation between amino acids during the translation. It ensures the proper alignment of mRNA and ribosomes during the protein synthesis and catalyzes the formation of peptide bonds between the two aligned amino acids during the protein synthesis. rRNA has a unique three-dimensional shape formed by interloops, and the structure creates a specific sites within the ribosome, that is A site, P site, and E site. A site is the amino acyl site, P is the peptidyl site, and E is the exit site. In case of prokaryotes, the large subunit is 50S and the small subunit is 30S. In case of eukaryotes, the large subunit is 60S, that is Fedberg unit, and the small subunit is 40S. So if we talk about the small unit, small subunit, in prokaryotes it is 16 Swedberg. In case of eukaryotes, it is 18 Swedberg. If we talk about the large subunit, that is eukaryotes have 23 Swedberg, 28 Swedberg, and prokaryotes have 23 Swedberg. So rRNA has a complex secondary structure characterized by formation of helices, loops, and other structural elements. These secondary structures are crucial for the proper functioning of ribosomes during the protein synthesis. So, prokaryotic cells typically have fewer sets of genes, but in case of eukaryotes, there are 50 to 55,000 sets of rRNA genes, that is equal to 10 million ribosomes in one cell. tRNA, that is transfer RNA. It is non-coding RNA molecule that carries amino acids to ribosomes from growing peptide chain mRNA nucleotide sequence. tRNA are coded by short molecules of 70 to 90 nucleotides. The set of three nucleotides on mRNA is known as codon, and the corresponding sequence is anticodon. The base pairing of codon and the anticodon forms a translation mechanism. Here in the structure, we can see it is the D arm, it is the T arm, it is the acceptor arm, and it is the anticodon loop. And the small structure here is the variable loop. This is the 2D structure and this is the 3D structure. So D arm helps in stabilizing the structure of RNA and influences the kinetics and accuracy of translation at ribosome. At the end of tRNA 3' hydroxyl base, there is an anticodon amino acid sequence that is attached, linking to the ribosomes to form a peptide bond. In case of tR, influences the tRNA effect on the translation by interaction with the ribosomes. The acceptor arm contains the 5' end of the tRNA made up of 7 to 9 nucleotides on the opposite end of the molecule base pairing. Here, codons in the anticodon loop, it is recognized by amino acid tRNA synthetase is paired to mRNA, and it determines the amino acid that is attached to the acceptor arm. So, thank you for watching.